In order to replicate DNA, you are making and breaking a lot of chemical bonds, and those are chemical reactions, and therefore you need enzymes and some other molecules to help you out. So what sorts of enzymes do we need? Well, first of all, you need an enzyme called a helicase. And we have a double helix, right? Two strands of DNA bound together by hydrogen bonds and twisting in a helix. Helicase unzips them and breaks them apart, essentially. Then you have single-strand binding proteins. These are not enzymes, but they do have an affinity to DNA, so they'll bind with that DNA and essentially keep it from just snapping back together, because obviously these nucleotides and these nucleotides like to join together. That's why they're in that double strand in the first place. So this sort of binds to these DNA molecules temporarily, keeps them from snapping back together. Also, as you are untwisting, I don't know if you've ever untwisted a thread, right? Or like a piece of yarn's got a bunch of threads together, and as you pull them apart, sometimes they get really knotted further back, right? This is an experiment. You could do it at home, right? Find some yarn and try to untangle it, and just, if you're not too careful and you just yank them apart, it'll get knotted really tight on the other end. You want to prevent your DNA from doing that. You don't want it to get over-twisted back here because of the untwisting here. And so topoisomerase is an enzyme that helps prevent that. All of this allows for another enzyme that's not on this image called DNA polymerase to add new nucleotides here to make a polymer of DNA. That's what DNA polymerase does. But DNA polymerase, while it is a fantastic molecule that can put nucleotides together into a molecule of DNA, into a polymer of DNA, cannot just start a molecule. It can only add nucleotides to an existing molecule. And so you have to have what's called a primer to start with. And a primer is usually made of RNA, but it will bind temporarily to this DNA here. And then DNA polymerase can add DNA nucleotides to this primer, right? To add the primer, you have to have another enzyme called primase. So again, primase adds primer. DNA polymerase will add DNA nucleotides to the primer. And this can only happen if helicases unzipped things, single-strand binding proteins have prevented these from reattaching, and topoisomerase prevents us from over-twisting in the back. Well, every time we add a nucleotide to this chain, right, we're binding those together chemically. We're making a covalent bond, and that is a chemical reaction, and we're building a larger molecule from a smaller molecule. Those are generally endergonic reactions, and so they require energy going forward. Where do we get that energy? Well, it turns out these nucleotides that are floating around have been kind of juiced up, and they have extra phosphates on them, right? And they are called nucleoside triphosphates, right? Nucleoside triphosphates. And, you know, a nucleotide normally is going to have one phosphate, right? Phosphate sugar backbone, but it has two extras, and those two extras get cracked off as you are putting the nucleotide on there, and they float away, and that breaking provides the energy necessary to add that particular nucleotide to the growing chain of DNA. Now, you may recall from our discussion of Watson and Crick that our double-stranded DNA is anti-parallel, which means those two strands run in opposite directions, right? And we mentioned that DNA strands run from 5' to 3', and you can only add nucleotides to the 3' end. So if my elbow is 5' and my hand is 3', right, the other strand is going to be like this, right? Its 5' is going to be over here, and its 3' is going to be over here, right? So you would add in opposite directions. Sorry, that may not be helpful, right? But you can see that here, right, we have a 5' and a 3' over here, right? 5' and a 3' over here. So as you unzip them, right, this new strand that you're adding here, right, starting with this primer, the existing strand is 5' to 3', so it's going to have its 5' over here and its 3' toward the replication fork, which is great because as you continue to unzip, you can keep adding new nucleotides at this 3' end, right? Once you have a single primer, you can just add nucleotides all the way in, and this will continue to unspool, and this will continue to add nucleotides on the way in. That said, this strand here, right, it's 5' to 3', which means 5' is here and 3' is this way, which means we have to add nucleotides that way. All right, well, that makes it much, much more difficult, right? So we end up having to add them in segments. So we add until, boom, we hit something where we've added before, and then we have to add another primer, then add nucleotides, boom, till we hit the primer we've done before, then go back, start with a new primer, add nucleotides, boom. And whereas this other time we had one primer, we can continue going and adding to that 3' end towards the replication fork. Here we're going away from the replication fork, so we have to add in what are called Okazaki fragments. And this strand up here is often called the leading strand. This strand down here is called the lagging strand. Lagging strand is done in fragments. So let's look at this. Imagine this is your replication fork over here. So you really want to be going that way with your new nucleotides, but you can't because our new strand is 5' to 3' in this direction. So we add primers here, and we add nucleotides in this direction. Boom, adding new nucleotides until we bump into whatever we've made before, right? And then we have to go back, right? Add a new primer, add more nucleotides, boom, until we bump into this, right? And then we have to go back and do it all over again. We have to continue adding these new fragments. We also have to take this primer out, this extra primer, and replace it with DNA nucleotides. We have a different kind of DNA polymerase that can do that, but that's fine, because once we take these out, DNA polymerase is good at adding nucleotides to an existing nucleotide. So that's pretty easy, but it's a little inefficient, right, because we're doing this in fragments, right? So here is our lagging strand, boom, 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 right? We're adding in fragments. Here's our leading strand, right? Once we did a primer over here, we can continue adding continuously new nucleotides as we continue to unzip this with helicase, right? So again, leading strand, lagging strand. So I provided you guys with a number of videos and animations which show this much better than I'm going to be able to do this just speaking at a computer screen and looking at still images. But remember, as you unzip this, you're doing this with a helicase, right, which unzips. You will have single-strand binding proteins, which keep these separate, right? You'll have topoisomerase over here, not shown, which will keep this from over-twisting. And we have primase, which adds a primer, and then that primer allows us to add, so here's another primer, add nucleotides from that primer, because DNA polymerase, which is our other most important enzyme over here, cannot just start from nowhere. It has to add nucleotides to something that's there already. So you have to have that primer, that primase, this pink one, has added, and then we add nucleotides to it, boom, until we bump into what we've done before. Now we're going to be able to go up to this primer, add nucleotides, boom, until we bump into this. And then we'll keep doing that over and over and over again. And most of this is a complex, meaning it's all hooked together. Then you have to go back to these primers and take them out, because those are RNA, and add DNA nucleotides to that, right? And we have a different kind of DNA polymerase that does that. It's DNA polymerase 3 that does most of our nucleotide adding. I believe it's 2 that fixes the primers. But anyway, I would encourage you guys to go watch a video about that. I've provided those to you. Because of the structure of DNA and the efficacy of DNA polymerase, you are very, very accurate at copying your DNA. That said, just like in any process, there are errors, right? And DNA polymerase actually has a built-in mechanism to sort of feel out kind of bumps, right, behind it. Places where, like if you add a T to a T or a T to a C or something, it's not going to fit right. So there'll be a little bump, and it will get rid of it and replace it, boom, with the appropriate nucleotide. So it actually proofreads itself, which makes the error rate even lower. That said, there are still errors. And so you have to have other mechanisms of fixing those errors. And so we have a way of doing nucleotide excision repair, which means you cut out the offending nucleotide that shouldn't be there, right? We had a C on one side. Instead of a G on the other side, we put an A, right? We cut out that A. We put a G. That's called excision repair, and it requires DNA scissors. And enzymes that cut DNA scissors, are called nucleases. So this is a good example of that, right? So we have a mess up, right? And you can see it's got a little bump there. Nuclease cuts that out, and then we have a DNA polymerase that will come back in and repair that, right? And it will repair it based on what's here, right? Based on what's on that other strand. It's pretty handy. Now, that said, even with these multiple redundancies, you are still going to end up with some mutations there, and sometimes these mutations don't necessarily happen during replication, right? You can have damage that occurs from mutagens, things like X-rays or UV light or carcinogens of other sorts, things like that. Now, this low but not zero error rate is really, really important when it comes to evolution. So we tend to want to avoid errors, because errors could be, there are three options for an error in your DNA. It could be neutral, which means doesn't end up mattering at all. Either it's in a place where it doesn't code for proteins, or it switches a nucleotide into another one, but it still codes for the same amino acid because there's some redundancy there, or it codes for a similar amino acid that may not change the structure of the protein very much, right? Neutral. Or it could be good, right? Could make your enzyme better at doing its job, or it could become a different colored pigment that actually makes you better able to hide or something like that. Or it could be bad. And bad is actually much more likely than good, right? It could make an enzyme that's non-functional. So we try to avoid these, but in the long term, this gives us genetic diversity, which is the raw material by which evolution operates. And so this non-zero error rate is very, very important. Okay, I mentioned that DNA polymerase cannot start from nowhere and build a DNA molecule. It has to add from existing DNA, right? Or existing RNA. So in our case, we build an RNA primer that DNA can add to. And then that RNA primer is eliminated. Now, normally that's fine, because if we're in the middle of the DNA strand, we can just add DNA to this other Okazaki fragment, for instance, and we can fix that. But when you're on the ends of linear DNA molecules, so remember, this is not an issue for bacteria who have circular chromosomes, but we have linear chromosomes, so we have ends. If we have a primer on the end, and we always do every time we replicate DNA, that primer goes away, and we have no way of adding the nucleotides, because we can't add them to the 5' end, and there's nothing to add on to over here, so you just end up with nothing, right? And then the next time you replicate, okay, there's nothing to bind to over here, right? And so you can't place nucleotides over there, and so you end up that much shorter. And so every time you replicate, the ends of your DNA molecule get a little shorter, and a little shorter, and a little shorter. So that's not good, right? We don't want that we don't want to lose coding DNA. So what do? Well, we add little bits at the ends of molecules, and this is an image that shows them through fluorescence, called telomeres. And these are essentially just useless DNA at the end of your chromosomes that we can lose, and it's not that big of a deal. They kind of operate like those, they're called aglets, right? Those things at the end of your shoelaces that protect your shoelaces from coming undone. That's what our telomeres do. These are called telomeres. One of the things that may cause aging is us losing our telomeres. Cancer cells have an enzyme called telomerase that rebuilds them, and so cancer cells essentially can go on to live forever. I'm not sure if you've ever read The Immortal Life of Henrietta Lacks or watched the Oprah movie, but it's worth doing. As we've already discussed, you have a lot of DNA in all of your cells, and it's made up of not just DNA, but your chromosomes are actually DNA around histone proteins. And the whole thing, the DNA and the protein, is called a nucleosome, and then it gets wrapped into a big condensed chromosome. And during mitosis, it's condensed a lot like this, but normally it's not condensed. It's this loosey-goosey spaghetti stuff. The reason for this is that you can't read a closed book. You can't read coiled DNA. And so we can turn genes off by coiling them. We call this heterochromatin, and open them up, make them useful by unrolling them. We call that euchromatin. We'll talk about how we use this to regulate DNA expression next video.